I have my email turned off so it won't make noises at us. Okay, so today we're going to start talking about sort of larger picture, larger scale aspects of microbiology. Remember at the beginning of the course we started with very structural aspects of microbial cells and all the components that make up the outside of the cell, and we went inside the cell, and then we started looking at how these cells interact with other organisms, like higher eukaryotic organisms. And now we're going to talk about how these organisms interact as communities on a global scale. But first, we're going to talk a little bit about microbial evolution. And again, to remind you that IEVAL is available. You might be able to there. But we're going to talk now about microbial evolution. Okay, so there's evidence in the fossil record that bacteria were present on planet Earth a very long time ago. Okay, so that's probably not surprising to you. But some of that is still evident in today's microbial ecology. Okay, so you can see here that there are things called stromatolites. So on the left, those are modern-day stromatolites, and those are bacterial communities that stratify according to who's in the community and also the things they're secreting. So calcium carbonate, for example, in these sort of sedimentary layers. And this is something that you can go and see right now if you wanted to. But then if you look back into ancient stromatolite rocks, you can see the same striations. And based on their carbon footprint, we know that they're microbial in origin. And so basically you can see this calcium carbonate that these microbes are secreting, and that's visible as sedimentary rock. Okay, so this again is just another image of some more stromatolites. I think they're very interesting. So this is modern day stromatolites that you can go and see in Australia, I believe. I think it's Australia, I'm not sure. And then over here, again, is another fossil that you can see, and we can date it back to 3.5 billion years ago, that this same sort of biological processes that were happening then are still happening now. And so that gives us good supporting evidence that microbes really were the first organisms on the planet, and that they were doing very similar biochemical processes that they do today. Of course, we're going to talk a little bit about the origins of life, because you can't really escape that based on what I just showed you. And before these cells could evolve into these microorganisms that we can see in the fossil footprint, there had to have a certain number of criteria had to have been met, okay? So you need the essential elements that you need to make organic molecules like carbon, oxygen. You need a continual source of energy for planet Earth, that is the sun. And then you need a temperature range permitting liquid water. So it can't be too cold and it can't be too hot, because otherwise the water would be frozen or it would be gaseous. Okay, so what are some other pieces of geologic evidence for early life? So basically just fossil evidence. So we see the stromatolites, like I talked about. But we also have evidence of microfossils that we can see, and I'll show you some pictures of those in just a minute, that look very similar to algae and fungi and some bacteria that we know that we can see today. We also can see biological signatures in the chemistry of these fossils, and so that indicates to us via isotope ratios and also cyanobacterial haploids, which I'll talk about in just a minute. But the isotope ratios, there's a certain ratio that dictates or signifies that there has been biological activity in that environment. Okay, so the elements in that environment have been altered in some way because of some biological processes, and you can see that in the isotope ratios. Haploids, which we didn't really talk about in the beginning, but they were on the slide in case you were paying attention a very long time ago when we were talking about cell envelope structure. So these are structural things that are kind of like steroids in the membranes of prokaryotes. And they provide structure and rigidity, and they're very durable. Okay, so that's why they're there. And we can see these in the fossil evidence that we can look at today. And those definitely come from prokaryotes. Okay, so if you look here, you can see on the left some fossilized microbes, and on the right you see your modern day microbes. And so on the left you can see some things that look very much like filamentous prokaryotes. Okay, so bacteria that can grow in long strings. And on the right is a modern day organism that looks just like that. On the left here in the second panel is a cyanobacterial colony. Looks very similar to cyanobacteria we can find in the ocean today. These are our photosynthesizing bacteria. On the left, you can see this fossilized algae, which is a eukaryotic organism, or a fungus, algae, and it looks very similar to that red algae that you can see in the ocean today. And so this is how the fossil record compares to modern day organisms. So all of that gives us really strong evidence that microbes were here a really, really long time ago. So that was the point of all of those slides. But how did they begin? Okay, so there's a couple of different models for how the origins of life started. And no one model really explains everything. So it's likely a combination of these three models that we're going to talk about, and maybe some other processes that we haven't yet thought of or discovered. Okay, the three we're going to talk about are the prebiotic soup model. Okay, so models are just things that we have a lot of evidence that allow us to structure a model. But a model is just a model, right? It needs to be tested, and all the parts of that model can be tested and proven or disproven, right? So these are models. And there's the metabolist model, and then the RNA world model. But everything started with RNA rather than DNA. Okay, so the prebiotic soup, what is that? So this is the idea that small organic molecules that make up biological cells as we know them today actually arose abiotically. Okay, so without cellular life available, these things sort of spontaneously arose. And how they did that was there were chemicals floating around in the early Earth, and there was a lot of pressure, and then a lot of electrical discharge, so like lightning, for example. Okay, a lot of lightning strikes. And because of that, the chemistry, when you apply heat and pressure and then strike it with lightning, that can cause chemicals, simple molecules, to complex together into what we think are now these larger biomolecules like glycine, for example. Okay? And to further support this model, you can actually do this in the laboratory. So there are some scientists who have replicated this in the lab by having all of these smaller complexity molecules and then applying electricity to them, and then out comes glycine, okay, which we know is one of the amino acids that help build cells today. And so this is in support of that model, that you can produce complex macromolecules from smaller molecules by applying electricity. And then it was thought that membrane compartmentalization of these complex molecules just spontaneously happen through mycel formation. Okay, so that will happen if you have a lipid bilayer. It likes to just make a mycel, and in doing that, it would capture some of these large macromolecules, and that is the model for how cells began for the prebiotic soup model. Okay, the metabolist model is that, again, some abiotic chemical reactions that were self-sustaining. So these were cyclical things. They gave rise to more complex macromolecules that then fed into early cells. Okay, so you can see that here. So this would be on the left, this cycle here of all of these chemical reactions that I'm not going to ask you to memorize, but you can see that one of the components is pyruvic acid, which could feed into the TCA cycle as pyruvate. Oops, sorry. And which would lead to further complex macromolecules, okay, and eventually amino acids. And so that is basically what the metabolist model is that these abiotic chemical reactions form the basis of cellular metabolism by having these byproducts that spin off these reactions that just run basically by themselves. that RNA was the first genetic code, or the first genetic acid that was available to cells. So we can see hallmarks of this in today's modern day cells, okay? So remember when we talked about DNA replication, and that DNA polymerase cannot work de novo. It cannot start assembling 5' and 3' without a 3' end to add onto. So you need an RNA primer there to do that. RNA polymerase can do that. It can start, creating, elongating strands of RNA based on a single base. Okay, so that RNA is still evident in today's cells in that the fact you still need a little bit of RNA during that DNA replication cycle. Okay. There's also evidence because we see that some viruses utilize RNA as their genomes exclusively. So there's double-stranded RNA viruses, there's single-stranded RNA viruses, there's some viruses that use RNA as an intermediate. And so it's very important to those entities as part of their genome and of their life cycle. And RNA actually has catalytic activity. So the peptidyltransferase activity that's in the ribosome that transfers the peptides in the growing chain of the protein, that's RNA. That's not a protein that does that. So RNA can have catalytic activity that can be very important to biological processes. We also know that we talked about small RNAs regulating gene expression, and so they do have a regulatory component to them. And then I already talked about how it's involved in synthesizing proteins. And that the ribosome, remember, is actually mostly made of RNA. And so all of these three models explain parts of how we got to modern day cells. And like I said, no model explains everything. And so it's likely a combination of these three that explain the origins of life. Okay, so moving on from that. So life has begun, okay? So we have cells that we can look at in the fossil record, and we have cells that we can study today that we can go out into the environment and look at. And we always want to know, as humans, how do things relate to one another, and how can we categorize them, okay? So what is the best way to do that besides just looking at things? So we already talked about prokaryotes. They look very similar to one another. So gram-negative rod-shaped is a very common way that bacteria look. So morphology is not very helpful in terms of organizing and understanding how bacteria relate to one another. So you got to look further into the cell. You got to look deeper in there. And how do you do that? Okay, so one good way is to look at the genetic material and specific parts of the genetic material to begin to understand how things, how closely related or how distantly related things are to one another. Okay, when I say things, I mean species. So you can look at a DNA sequence, and you can have, if you're building a phylogenetic tree, so right here, this is a very simple phylogenetic tree. You can have a root sequence, so that's the sequence that is your root. And then you can look at other sequences, for example, 2A and 2B, that would be based on a round of DNA replication, or likely many, many rounds of DNA replication, where these mutations occur. And you can see that they look very similar to the root sequence, but slightly different in some of the bases. And then if you were to do a third round, you would see that that sequence has diverged, okay? So sequence 2 is more closely related to the root than sequence 3 is. And that's just a very simplified way of looking at phylogenetic trees, okay? It's actually much more complicated than that. And what you need in order to do this, to construct a phylogenetic tree that you can have confidence in, you need what's called a molecular clock. And so that's something internal to those cells that is sort of keeping time as a chronometer of when those species diverge from one another. Okay, so you need something that you can compare among all of those cells or all those species. Okay, so these are all the criteria for a good molecular clock or good chronometer. And so first of all, it has to be a universal molecule, which is logical, right? All of the cells need to have that molecule. Okay. It has to have a conserved function in all the organisms, so selective pressure or natural selection pressure on that particular entity is the same across the all of the species, okay? You have to be able to align them together so you can look at them and say, oh, here if they look like they are very closely related or they're not. You need to have a constant substitution rate so that sequence divergence is proportional to time. Okay, so that is one of the most important criteria. It has to be slow evolving, not something that's prone to like high rates of mutagenesis. Okay, because then you would get erroneous results. So it has to be something that every organism has. It has a conserved function in all those organisms. You need to be able to align the molecules. It needs a constant substitution rate. And then also, it has to be strictly vertically transferred. So from parent cell to daughter cells. None of this horizontal gene transfer can apply to your molecular chronometer because that will skew your results. Okay? So it has to be something that is inherited vertically and not horizontally. Okay? So after all of this, we find some molecules in cells that fit these criteria. And all of them are nucleic acid based, okay? And they're DNA based. And so you're looking at the 16S ribosomal DNA gene, okay, as a good one. And that's what we're going to talk about here. There are some others that can be used, but in the context of this class, we're going to talk about the 16S ribosomal DNA. So we know what the 16S ribosomal RNA is, right? That's the piece of the ribosome, okay? That is the thing that binds to the Shine-Dalgarno site, right? But it has to come from somewhere. So it is encoded by RNA polymerase, or transcribed by RNA polymerase from the DNA. So I'm not talking about the RNA molecule here. I'm talking about the DNA from which that 16S ribosomal RNA came from. Okay? So it's not a messenger RNA that is transcribed from that. It's a structural component of the ribosome called the 16S ribosomal RNA. Everybody got that? It's the DNA, okay, not the RNA. So it's just like, if you think of it as like a gene, but it's not really a gene, okay? Because it doesn't go on, it is a gene, but it doesn't go on to encode a protein. It just encodes an RNA, okay? So the most widely used one, like I mentioned, is the 16S ribosomal RNA, or we're going to call it the 16S ribosomal DNA. Okay, and that fits all our criteria because it is nucleic acid sequence that you can take and look at in your A's, T's, G's, and C's, and then you can align that with other 16S ribosomal DNA genes that you have gotten from other species, and how closely related they are in sequence means that they're very closely related how closely related in how they relate to one another, and if it's further that more distantly related, then that sequence will look different, okay? So this is what 16S ribosomal RNA looks like. Okay, it has some secondary structures to it. And I just want to remind you again that it's a structural component of the ribosome. And it also has these really nice convenient features. Okay, so it's about 1.5 kb in length. So that happens to be a piece of DNA that is very easy to amplify by PCR. It's not too large, it's not too small. It's just a nice piece of length of DNA. So 1.5 kb in length. Okay. It has conserved regions across all life forms. Okay, so that means when you're looking at groups of cells, then you can be confident that they all have this. So that's convenient. But it does have some variable regions, okay? So that you also want it to be slow evolving, right? Like you want it to have evolved somewhat, so then you can be able to tell in this, this one is highly divergent. That means they evolved away from each other a long time ago. Or they're not divergent, and that means they didn't evolve away from each other that long ago. And they're very closely related, okay? So how does this work? Very simple. So you have your cells, okay? So if you're trying to do this in the laboratory, you can easily just start with a single colony of organisms, inoculate your culture, grow them up so you know that you have the same species in that culture tube. Then you're going to isolate its DNA. So you have purified DNA prep from those cells. And then you're going to do PCR. And your PCR primers are going to amplify the 16S rDNA gene. Okay? And then from that, you're going to sequence it, or likely you're going to send it to a core and they send you back this computer file that has all of your A's, T's, G's, and C's in it, and then you can look at it, right? And so you can do this. We'll start this again with a different batch of cells that was potentially different species. And you can do that, as many times as you want. And then in this case, we have 3 examples. So just imagine that we started with 3 different batches of cells, and we're trying to determine how closely related they are to one another. And so we extract their DNA, we do PCR on their 16S ribosomal DNA gene, and then we use the computer to align all of that sequence information that we got from that process. And then we can see how we can create a phylogenetic tree. And we can see that species 1 is more related to species 3 than species 3 is related to species 2. Okay? And it's all based on interpreting the number of differences in the base pairs of your aligned sequences. Okay. So this is how the archaea were broken out into another domain of life. Okay, so not too long ago, they were grouped with the bacteria because they looked like bacteria. They had all sort of hallmarks of bacteria. They lived in different environments than eukaryotes normally would, and they were just sort of lumped in there. But after doing all this molecular work, Carl Woese is the one who came up with this idea of using the 16S ribosomal DNA, and then he set out to look at all of these organisms and compare them to one another, and he actually said, these archaea do not belong to the bacteria at all. They're actually more closely related to eukaryotes than they are to the bacteria, and they need to be in their own domain of life. And so that's how we did that. That's through the 16S ribosomal DNA. Okay, so I'm just going to add in one thing here. So this is based on taking a single species, extracting its DNA, and then doing all of this process, and then you have your sequence, one sequence, you can align it. All right. I just want to give you a hint of what we're going to talk about in the microbial ecology lectures, is that you can do this on an environmental sample for which you don't know what's in there, and you haven't grown them. You're just extracting them from the environment, take their DNA, and do the same PCR using universal primers, okay, and amplifying all of the ribosomal DNAs at once. Okay, so that is next generation sequencing. So that's why we call that high throughput sequencing, because you can sequence all of that at once, and then using some pretty complicated computational methods, you can organize all those together. And you can say, this community looks similar to this community, or it looks different from this community. So you can do it at a higher level, right? So I just want to give you the seed of that. We're going to talk about that next time, but that's kind of where we are with this 16S ribosomal DNA. It's called amplifier-based sequencing, because you're amplifying something with PCR. And I did forget to mention that it's very convenient that you can use universal primers. Okay, so that's another criteria, is that the sequence is so close in all of the organisms that you can use the same set of PCR primers for every single organism to amplify the 16S ribosomal DNA. So that if you were doing this in the lab, you don't have to keep redesigning your primers for every species, right? So you can use the same set of primers. Yes?
Maybe there's an obvious answer to this question, but I got a little confused when you kept saying like 16S ribosomal DNA gene, but it's saying 16S rRNA.
Yeah, so she's asking again about the clarification between the 16S ribosomal DNA and the 16S ribosomal RNA. So they're kind of used interchangeably, but what you're amplifying is the DNA.
Okay.
Yeah. But if you look at the textbook and the literature, they will use those terms interchangeably. But what you're doing is targeting the DNA. Okay. So, moving on. So that was all about vertically transferred pieces of nucleic acid, right? That is on the chromosome, and as the cells are replicating and they're duplicating their chromosome and it's getting deposited into the daughter cells, that's vertical transfer. With prokaryotes, we have this issue of horizontal gene transfer that we've talked about many times, and that's where cells can just pass DNA to to one another without having undergone any cell division. And so this complicates phylogenetic analyses with prokaryotes, because sometimes things that really are unique to that organism were obtained through horizontal gene transfer, and it makes them inherently different from their brother that's very closely related to them, which if you use the 16S ribosomal DNA method, you would say, okay, these two things are the same species, and they didn't diverge from one another very long ago. But one of them could be E. coli that you can find, in your gut, and it's fine, and it's not causing a problem. The other one could be E. coli H1N157, which has a toxin, and it causes severe diarrheal disease and is very problematic. And it acquired that through horizontal gene transfer. Okay, so they can do that by obtaining plasmids that have particular genes on them. You can have transposable elements that come in, or you can have bacteriophage carrying DNA around from bacteria to bacteria, and you can look at that. If you remember that a strong sign of horizontal gene transfer is the GC content. Okay, so if you're scanning the genome of an organism and has 50% GC content, and suddenly there's a dip in the GC content of that particular region of the chromosome, you know that it came in from a foreigner. Okay, so horizontal gene transfer is when the bacteria are taking DNA from their environment, and GC content is a very good and reliable way to show that one species is the same as another species. Okay, that's just the overall percentage of G's and C's in relation to the rest of the nucleic acids. Okay, so for example, if you have 50% G's and C's, then the other 50% are A's and T's. So if you're scanning along the genome and it's 50%, and then suddenly it's 40%, that means that piece or segment of DNA came from an organism that had 40% GC content and 60% AT content. Okay, so it just signals to you as a scientist like, hey, this came from somewhere else. And why do we care about those? Because a lot of times, because these things are maintained in the population, they offer some selective benefit. Okay, so these can be described as pathogenicity islands or genomic islands, and it just means that it's large sets of genes together, sometimes operons, sometimes individual genes, on these chunks of DNA that have come from a foreigner. Okay, so they're transferred as a unit, and you can see that when you are doing chromosome analysis of bacteria. And our example was E. coli 157H7, and other examples could be for when we talked about, we didn't talk about that. We talked about Agrobacterium tumefaciens, which is a bacterium that causes galls in plants. We know that Agrobacterium tumefaciens that doesn't have the TI plasmid looks very similar at the phylogenetic level, just looking at their chromosomes, than one that has the TI plasmid. We know that they have very different phenotypes in terms of virulence. This one causes disease, and this one doesn't. And so this is sort of a conundrum for people who are studying systematics of bacteria and trying to group them, like really hard, trying to group them into groups, but then you have these outliers that do all these crazy things, cause problems in humans or cause problems in plants, and we don't know exactly how to characterize them. So if you look at the taxonomy of bacteria, you can sometimes see that's a little bit of a mess, and that people are constantly moving things around from species to species, renaming them, sometimes taking them out of one genus, putting them in another, and writing a manuscript to describe that, and then someone will come back and say, no, actually it needs to belong back here, and we didn't like that you moved it there, and so the names are constantly changing. So, for example, in my graduate class that I'm teaching now, I have to constantly remind myself that Erwinia keratodra is not called that anymore. That's how I learned it. Now they call it Dickeya dedantii, and so it's like, okay, I have to remember that it's called that and not something else. The other example is Agrobacterium tumefaciens, which is a big pathogen. There were some scientists who wanted to group it with Rhizobium, which is a beneficial species, okay? Or genus that contains beneficial species that are involved in nitrogen fixation. They wrote a manuscript and then moved it over to that genus, and then there was such such an uproar from the scientific community that 150 different scientists signed this memo of trying to say that no, we do not like this. We want it to be called Agrobacterium tumefaciens because it's a pathogen, and if you're grouping it with these beneficial organisms, it's going to confuse everyone. And so then they moved it back. And so horizontal gene transfer is a problem for systematics of bacteria. I just kind of went on a long rant to say that, but it's kind of frustrating when you're not into systematics and then the names keep bouncing around. But I understand that why it's important. Okay, so it's important because some of these things are subject to quarantine regulations, for example, so it matters, the name, right? So it matters if you've named it the right thing because that means that particular sample that has that organism is either allowed or not allowed in a particular country. It matters when you're doing diagnostics because you can say, oh, I have this bacterium, or actually, no, I don't have that because it's named something different and it's been declassified as that. And so the naming is important, and I'm not trying to say that it's not. It just can be sometimes frustrating when you're not in charge of the naming. Okay, so we're talking about microbial evolution and how, all the way from the origin of cells and how cells can evolve and how we can tell if they have evolved from one another. But there's also this theory, endosymbiont theory, that we touched on in the first part of the course, and you probably have maybe heard of in your other biology classes, where bacteria that were bacteria or photosynthesizing bacteria actually invaded eukaryotic cells and then began to be so comfortable with them, and they relied on each other so much that they became the organelles that we know of today, like mitochondria and the chloroplasts in plant cells or animal cells. Okay, and so we can see that, evidence of that today. So there are some organisms, chlorella species, that live inside these paramecium, which are eukaryotes. The paramecium provides shelter for the algae, and the algae is providing photosynthate. Okay, so you can see how this symbiotic relationship can evolve. Okay, and so this was Lynn Margulis, who's at the University of Massachusetts Amherst, and she came up with this endosymbiont theory that the mitochondria and the chloroplasts were actually bacterial in origin. And this was not without controversy. Okay, so she was proposing this, and many people were saying, no way, no, this can't happen. This sort of bucks the system and is going against what we know of and what we describe in terms of evolution. Okay, but she had all this evidence, and she said, but look, guys, this is what it is. Okay, so if you can see in the chloroplast and the mitochondria, there's a double membrane here, that electron transport components that we see in microbial cells are present in mitochondria, and so it really looks similar to them. Some of the phytosynthetic complexes that we see in chloroplasts are very similar to what we see in cyanobacteria. So, here's my evidence for that. They behave like endosymbiotic organisms, so they're living inside another cell. They're reproducing independent of the cell. Okay, so that is also evidence that these things were once separate from one another. And it's based on what she thought was extreme reductive evolution, is that the chloroplasts and the mitochondria that came from bacteria actually underwent reductive evolution in such that they lost so many genes that they are now reliant on these eukaryotic hosts and they can't live anywhere else. Okay? And so, the evidence that she was showing is that these actually have their own chromosomes. So they have circular chromosomes, very similar to bacterial chromosomes. And that they have ribosomes that look very similar to prokaryotic ribosomes. And then if you use our technique that we just talked about, which is our 16S ribosomal DNA or 16S ribosomal RNA, and compare them, these chloroplasts and mitochondria actually look very similar to bacteria, and they don't look similar at all to the eukaryotic organisms in which they inhabit. And another piece of evidence is that we know that antibiotics, some of them that target specific functions in prokaryotes, actually can harm mitochondria and chloroplast. Very similar to what we might expect for a eukaryotic system. Okay. And then I just remembered that I did not finish my lecture last time. I'm sorry about that. I'm going to go back and do that. Just a couple slides. So which one? I'm sorry, I'm like really confused. The one that had the ELISA, the diagnostic one?
23.
That was 22, right? Okay, so please correct me if I'm wrong. This quarter is kind of crazy. I'm teaching 2 classes. Did we go over this? Okay, all right. I remember correctly. Okay, so this was talking about different molecular techniques that we can use to identify organisms. Okay, so this would be an example of using PCR to try and figure out if our samples are contaminated with a specific strain of Clostridium botulinum. Okay, so there are different genes that involve different toxins. So the type A, type B, type E, and type F toxins in Clostridium botulinum. You can do what we call a multiplex PCR. So that means that you have different primer sets with different genes. Okay, so in this case we have 4 primer sets. So the type A, the type B, the type E, or the type F PCR. Okay, or type genes, and those you put all of those primers into the tube with your chromosomal DNA. Okay, and then you allow PCR to happen, and then you run your gel. Okay, and so if you have one product that is the size under type A, that means that your sample had the type A toxin. Type B is a different size, or amplicon. Type E is again a different size amplicon, and type F is a different size amplicon. So when you're running your samples and you have amplicons of the size of the type A, the type F, and the type B, you know that that particular strain had those 3 genes, okay? And that is something that would be diagnostic for the particular strain of Clostridium botulinum. Whereas the second sample here has type F, type E, and type A, and you can see that the sample, the first and the third sample, look very similar to one another, okay? And so that's what we call multiplex PCR. So that allows you to detect multiple genes at the same time in the same PCR tube. And so you can use that for diagnostics. Another thing that you can use in a clinical setting is serology. So this is based on enzyme-linked immunosorbent assay, or ELISA for short, which you've probably heard of before. And what it does is it's based on antibody recognition of an antigen. Okay, so it's basically using the immune system of an animal and capitalizing on that to detect a particular antigen. And so you'll have antigens from, one example of how you can do an ELISA is you can take patient serum, okay, and see if it has antibodies to the particular antigen from a virus. And how this is, the example is explained in your text, is looking for patients that might be infected with Ebola. Okay, so what you do in this case, and this is just one way of doing ELISA, there are other ways to set it up, but you would have a 96-well plate, and you're going to zoom into one well of the 96-well plate, and with that tube in that, or that well in that 96-well plate, you would have coated it with some known antigen of Ebola. Okay, so that's something that you put in there on purpose. You probably produced it in recombinant fashion, so you asked E. coli or some other organism to make it for you. So you have this purified protein, which you know is antigenic. So that means it stimulates an immune response in humans. Okay? So you're going to bind that to your wells, and then you're going to rinse it off, and anything unbound is going to leave, and everything that's bound will stay. Then you're going to take the patient's serum from their blood, which you separated into the serum, and then you're going to put that in the well. Okay? If that patient has produced antibodies to the Ebola antigen, it will bind to the antigen that is bound to the well of your plate. Okay, so you have this little stack that's forming here. Okay, then you're going to wash off any unbound serum, and any other antibodies that were not specific to the Ebola antigen will also get washed off. Okay, now you need to figure out how to see that, how to visualize that. So you take a secondary antibody that was raised in rabbits against human IgG, so it's rabbit anti-human IgG, okay? Because some poor rabbit has been injected with this and raised an immune response to it. And then, if you had some primary antibody from that patient in the serum that has bound to the Ebola antigen, that rabbit anti-human IgG antibody will bind to it. So you start to have this little stack, right? You have antigen, the primary antibody, and then you have the secondary antibody here. And then you wash off everything that was unbound. Then you add a substrate, which will turn a color if that secondary antibody is present, because it has a color-changing enzyme attached to it. And then it will turn either blue or yellow, depending on the substrate. And that will tell you that that patient actually has a viral load of Ebola in their serum. Okay, so that is a diagnostic test based on the principles of immunology. Okay, and that's it.